Hello everyone. Today we will learn about AIDS and HIV infection. Before we begin with HIV infection, definitely all of you must have heard about AIDS. So what is AIDS? AIDS is acquired immunodeficiency syndrome. That is, it is a collection of symptoms because of the underlying infections and malignancies, which basically results due to the infections caused by a virus which is called as HIV, that stands for human immunodeficiency virus. This infection is now a pandemic. Currently, we have almost 40 million people who have been living with HIV infection, and 25 million people have already died from this disease. So this is basically a global view of HIV infection. As per the recent statistics, we could see that almost the entire world is affected. More than one country, more than 10 countries have been affected by this infection. And the last stage of HIV infection is basically what we call as AIDS. So AIDS was first discovered in 1981 in case of homosexual drug addict males. When the doctors examined these drug addicts who were also homosexuals, they found that they had two things in common. And this was the beginning of the discovery of AIDS. These males had pneumocystis pneumonia infection and another situation called as Kaposi sarcoma. And later, these two conditions became the markers of a collapsed immune system, which is ultimately the AIDS. And only because of these situations, when the doctors examine that when any individual is affected from HIV and reaches the last stage, they lose their immune competence, they become more vulnerable to overwhelming and fatal infections, and finally they die. And so this condition was given the name called as AIDS. So this is a picture of how a pneumocystis pneumonia, which basically affects your lungs, and we have Kaposi sarcomas, you have red colored lesions, and these two are indicators that the individual has progressed to the last stage of HIV infection that is basically called as AIDS. So let us see what is HIV. What is this virus? So in 1986, this virus was given its name that stands for Human Immunodeficiency Virus by the ICTV, that is International Committee on Virus Nomenclature. This virus is very unique because this belongs to a group called as lentivirus subgroup, which comes under a group called as Retroviridae family. Retroviridae. What is this retroviridae? Retro means reverse, viridae means virus. So this virus belongs to a family Retroviridae because of presence of an enzyme called as reverse transcriptase in its genome. Another interesting thing is it belongs to a subgroup called as lentivirus. Lentivirus refers to slow virus. And the reason why they are called as slow viruses because these are known for latency. That means the virus remains hidden in your system without showing any symptoms. It has a persistent viremia. They're continuously present in the blood. They infect the nervous system and they make the host immune response very weak. And this virus has a very high affinity for a specialized type of cells in our body, which are called as CD4, T lymphocytes, and monocytes. Why is it interesting? Is it that these CD4 T lymphocytes are the initial defense mechanisms of our body. And we can imagine that if the first defense itself is compromised, itself is attacked by this virus, so the entire immune system becomes compromised. So we will see in detail what is the replication and structural characteristics of HIV and what is the clinical manifestations of HIV. So let us start first with the structural characteristics of HIV. Now, if we start with structural characteristic of HIV, the HIV is an spherical enveloped virus of about 90 to 120 nanometer in diameter. As you could see in the picture, it has an envelope, and outer to the envelope, it has an inner to the envelope, it has an icosahedral nuclear capsid shell, and inner to that, it has a cone-shaped core. So this is another interesting structural, seen in HIV is that it has an extra capsid shell. So it has a cone-shaped shell, and it could see, I could just highlight it. If you see that it has a cone-shaped cell, cone-shaped nucleocapsid, and then surrounding this nucleocapsid, it has another layer. This is what we said, that it has another extra capsid shell, and followed by it has an envelope. As I mentioned earlier, that it has a spherical envelope. The envelope contains a very unique protein called as GP120 and GP41, which is basically resulting from the cleavage of a G protein called as GP160. Apart from this, it also has another two enzymes, P32, P10, P55, and two copies of single-stranded RNA. So these are the few structural uniqueness of HIV. So the genome of HIV, which is basically your RNA, can be divided into two types of genes. One is structural genes and one is non-structural genes. The structure gene mainly codes for three genes: GAG gene, POL gene, and M gene. The GAG gene is basically genes which produces proteins which are responsible for the core, that is the nucleocapsid, and the shell of the virus. And in that, we have a precursor protein, which is P55, which gets cleaved by the enzyme protease. The virus uses the host protease enzymes to cleave this P55 into P17, P24, and P15. At this stage, the virus is usually not detected in our serum, but we can identify the presence of this virus by the presence of this particular main antigen, which is called as a major core antigen, which is called as P24. And this antigen is present from the early stage of the infection till the appearance of the virus. So this is one stage where we can detect whether the virus is present in our body or not. The second type of gene which we have in structural gene is called as a POL gene. POL refers to polymerase, or specifically called as the reverse transcriptase. As I said earlier, it is a gene. It's also recovered from a precursor protein, which is again cleaved by the protease enzyme. So the P64, which codes for many enzymes, it has reverse transcriptase, and then P51, which has only reverse transcriptase activity, and P10, which is a protease which cleaves the GAG, which we saw in the previous, and P32, which is an integrase. So the entire thing is combined from POL gene. So POL gene is basically referred as a precursor protein, out of which gets cleaved into P64, P51, P10, and P32. The third type of structural gene is this M gene. This engine is what we saw in the previous slide, where the GP160, that is a glycoprotein, GP stands for glycoprotein 160, which gets cleaved into GP120 and GP41. These are very crucial glycoproteins because these proteins are responsible for attachment of the HIV virus to our CD4 T lymphocytes and then penetration into the host cell. The second group of genes which we find in HIV is the non-structural and regulatory genes. So under non-structural regulatory genes, as the name suggests, they are more important during the viral replication, virus entry, virus exit, as well as the expression of the genes. So here we have VIF, viral infectivity factor genes, which refers to the effectivity of the virus particles to infect a host cell. And VPR refers to, which is referring to PR, that is promoter. So it refers to, V refers to viral, PR refers to promoter. So it's promoting the replication, transcription, and translation. VPU and VPX, these are two genes which are responsible for the maturation and release of the viruses from the cells. And we have two types of HIV, HIV-1 and HIV-2, and these genes are distinct in HIV-1 and HIV-2. And these genes are useful in distinguishing the type of infection which is caused, whether it is by HIV-1 or HIV-2. The next gene, which is we have TAT gene, which is also called as trans activating genes. T for trans, A for activating genes. And these are responsible for the synthesis of all the viral proteins. REV is RE is for regulator and V is for viral. So regulator of viral genes, which is again responsible for expression of the structural genes. NEF is a negative factor gene, which is responsible for the down regulation of the viral replication. And this gene is very important because this actually regulates the latency of the virus. So if this is more active, the gene is more active, the virus will be more latent. And the last important gene, which is a non-structural gene again, which also is one of the unique feature of HIV, is the long terminal repeat sequences. What do you mean by long terminal repeats? Long, the sequences are long. Terminal, they are present at the ends. And repeats, they will be the same sequences on both the sides. So as you can see in this picture, they are two long terminal repeats. They are terminal repeats present at both the 5' end and as the 3' end of the RNA genome, that is the HIV genome, and they both have the similar sequences. So they give both promoter, enhancer, and integration signals, and these are very useful in increasing the viral infectivity. So these were the structural regions of HIV. Let us see what are the variations seen in HIV. As I earlier said, HIV are of two types. We have HIV-1 and HIV-2. And interestingly, HIV-1 was the first one which was discovered. But later, after the discovery of HIV-2, we could find that HIV-2 was more severe than HIV-1. Although they have a lot of similarities, but it could find that it was less pathogenic. HIV-1 was less pathogenic than HIV-2. And HIV, the reason why we are not able to get vaccine is because HIV is a highly mutatable virus. So every now and then HIV keeps on mutating and it keeps on changing its nucleotide sequences. It prefers to change from cell to cell. That is what is cell tropism. It changes its growth characteristic features. It changes the cytopathological. And people and clinicians who have been isolating HIV from different races and persons, they also found that from the same person, the HIV taken up from different sites were also different. So this great variability accounts for the high mutation seen in this HIV. And the reason for this mutation is basically the error which is caused by the reverse transcriptase enzyme. As we all know that transcription is highly error prone compared to the replication of DNA. So as I mentioned, HIV-1 and HIV-2 and HIV strains have been classified into 10 subtypes based on their structural differences of their GAG gene. And these 10 subtypes have been designated as A to J, and they are considered come to be coming under group M for major, whereas few have been related isolated. They fall into the group outside group M, that is group major, and they have been designated as outlier group O. And few of them, after this discovery of this group, few more have been identified and they have been newly identified and they have been identified as group N for the new group. So let us see what is the mode of transcription. I know how most of you must be definitely knowing that HIV is transmitted mainly by the sexual contact. Not only sexual contact, whenever the virus enters the body through any infected body fluid. When we say body fluid, the body fluid can be infected cells, semen, blood, or any body fluid where the virus is able to transmit. So the body fluid could be even breast milk also, saliva also. So all these are body fluids which can be responsible for transmitting this virus from one person to another. And the primary most or the 75% of cases of HIV has been done, transmission is by sexual contact. So whenever a partner is having an infection of HIV and the partner is having intermediate intercourse with the healthy partner would be spreading this infection. The virus has been found to enter the body through the linings of the vagina, vulva, penis, rectum, or even mouth. So we all know that viruses are highly penetratable, they are highly smaller, and they are able to enter the body through all these body fluids. And people who are having already any sexually transmitted diseases, the bacterial transmitted diseases such as syphilis, Gonorrhea, genital herpes, or any other chlamydial infections, bacterial vaginitis, they all are most susceptible to acquire HIV infection, even if the HIV virus is not in their body. The second most common mode of transmission is through the parenteral route. Parenteral route is where we get the infection through blood transfusions or blood products. So somebody must have donated the blood and the blood might be infected with HIV, and when we get the blood samples, we may get the virus. Another way of getting these infections through the parenteral route is sharing of unsterilized needles or syringes, especially seen among drug addicts who share needles and syringes to share some drugs and which may be contaminated and they end up getting sick. There can be also some unfortunate instance where we may get some pricks, accidental needle pricks, or we may come in contact with contaminated fluids. We may get the infection. And has been found, as I said, they are also able to be transmitted through organ transplantation. So all these comes under the parenteral route. And the third route, which is another most common route, is the 15% or 30% of children who have been delivered by mothers. So this is called as vertical transmission, where mother who is already having HIV would be able to transmit these infections to their babies. So this is all about the transmission of the viruses. So in this way, we will also see the HIV multiplication and the clinical manifestations, diagnosis, cure, etc. Thank you.